Welcome, everybody. Thank you so much for joining us for this exciting webinar. Before I begin, my name is Rashmi. I'm the marketing manager at Protein Fluidics. I will be your moderator. A couple of topics of housekeeping before we begin. All your microphones are muted. You will be able to communicate with us via the chat. You should be able to see this on the right-hand side. And a quick overview of the agenda. We will first hear from Katya Nikolov and have a short pause with a few questions for her, and then go on to Dr. Margarite Matossian, and then you can ask questions to both speakers. So without further ado, I would like to introduce Dr. Katya Nikolov. She is the Senior Application Scientist here at Protein Fluidics. She has an MD from the Russian National Research Medical University. She has been a postdoc at the Memorial Sloan Kettering Cancer Center. He has a vast experience in working with 3D cell models and organoids. He has been a research fellow in New York and has worked on various startups. She has several publications and is a member of the American Association of Cancer Research. Without further ado, our Senior Application Scientist, Katya Nikolov.
Okay, well, thank you, Rashmi, for the introduction, and thank you, everyone, for joining the webinar today. So I will be talking about automated organoid assays using the microfluidic-based Puma system. First, I'd like to acknowledge our collaborators, Dr. Matthew Burrell's group at Tulane University. They work on developing triple-negative breast cancer patient-derived models. We also work closely with the team at Molecular Devices and with Oksana Serenko and Matthew Hammer. They help a lot with acquisition of confocal images and data analysis and many other assays that we do. So today I will talk about Puma system and how it works. I'll walk you on how to use organoid assay workflows with Puma system. And I'll show you an example of the data and assays that we use to do multifunctional profiling of 3D cell models. Now, why we've decided to build the system like that? These days, we witness really shift towards the more physiological 3D models for drug discovery. And when we talk about cell models and drug development, the most common thing that pops into mind is screening of large compounds of large libraries of compounds. And these screens are typically focused on really analyzing large number of samples, but with low outcome data points. Now, on the other side of the spectrum, there are many interesting and sophisticated organ-on-a-chip devices, but usually they would use few, maybe one or two samples, organoid samples, but they allow to produce really large number of data points and data. So essentially, here we kind of move from really simple model system to a very complex model system. And the Puma system that we designed, we think really fits nicely into this sweet middle spot when you can do medium scale screens in automated fashion and do what we like to call multifunctional profiling of your model. Now, our vision is really to have easy-to-use open lab on a chip for doing 3D assays where you can maintain physiological conditions. The system that would accommodate really different types of organoids. They could range in size between 100 microns to 1 millimeter. If needed, you can use some matrix for these assays. And we automated all complex assay steps for you, which really allows spend less time, less reagents on running complex assays. It allows to minimize handling errors and get really high quality and reproducible data with your precious organoid samples. Now here I mapped out a workflow that we developed and use with the system. The first step would be to load your sample, organoid or spheroid, in the Flowchip. Then the chip is placed inside the machine, and machine itself is sitting inside the cell culture incubator. So essentially, you run all your assays under standard cell culture conditions. Then you have to select the protocol, which is preloaded in the machine that you would like to run. Protocol is completed, you take your plate out, and you can use it for microscopy. It is possible to do fluorescence microscopy. You can do also luminescence-based readout. You can do plate reader-based downstream assays. And if needed, we can collect samples also for downstream analysis, for example, for metabolites assessments. Now, the heart of the system is the Flowchip. Here you can see the frame with 4 Flowchips in it, and you can use in your assays between 1 and 4, depending on the needs. So each Flowchip has 8 test lanes, and each lane is designated to 1 sample. So essentially, you can do between 8 and 32 samples. Again, depends on how would you like to do your assay. Now, in each lane, there's a sample well where the organoid or spheroid is positioned, and the sample well is connected to a number of reservoir wells through microfluidic channels. And these reservoir wells, you can fill up with anything, like, for example, medium with compounds. It could be different washing buffers. It could be staining buffers. They can be used for collecting supernatants. So really highly customizable depending on the configuration of the assay that you would like to run. Now, the well itself is pretty small. The volume of the well is 20 microliters. And you can see here that your sample is positioned at the very bottom in the protected sample chamber, what we call it. The diameter of the protected sample is about 1. It is optically suitable for doing high resolution imaging. And overall, the dimensions of this frame and the flow chip resembles standard 384 well plate with just minor modifications. So this plate is absolutely suitable and compatible with all instrumentation like standard plate readers or microscope or high content imagers. Now, like I said, the protected sample chamber is our key proprietary technology. So here you can see how the sample organoid is loaded into the well. And again, it is positioned inside this protected chamber. We can do fluid exchange from the channels, and we can do up to 95% exchange. The volume of this protected chamber is about 1 microliter. Now, if there's a requirement to, for example, drain your well completely and remove medium completely, you can do so by doing it from the bottom of the chip. Now, when the flow chip is loaded, sample is there, and all reagents are loaded, you are ready to start your assay. The interface is really simple to use. You just have to select the protocol, and the list of protocols is already preloaded on the machine. And then just you click the run button and kind of walk away and let the machine do all the steps of the assay for you. Now, let's take a closer look what's happening inside the flow chip while the assay is running. So here you can see an example, an outline of the flow chip with the sample. First thing that we can do, for example, we can remove old medium and replace it with fresh medium. Let the sample incubate for desired amount of time. Then we can remove this medium and transfer compounds. Let the sample be treated with the compounds. Then we can collect supernatant back in this well and transfer staining solution. Let our sample stain, remove stains, and do a wash step if needed. Now, at this point, when all the transfers and sample collection is completed, your assay is done, and the plate is ready to use for the downstream analysis. Now, you can really use different kind of samples with the machine. It could be single cell spheroids formed from standard cell lines. It could be tumoroids developed from primary tumors. We also did assays with primary and iPSC-derived organoids. Now, organoids typically require matrix for culture, and you can use matrigel with the system. It also works very nicely. So here, I would like to show you an example of how we do multifunctional profiling. This study was done using breast cancer patient-derived tumoroids that were loaded in the plate and treated with different compounds. After that, we did viability test assessment with stain for live dead, these tumoroids, and did confocal imaging. Also, from the same samples, we collected supernatant and later did downstream analysis of metabolites. And also at the end, we did immunofluorescence staining for biomarkers and again, did confocal imaging for that. So you can really see here there are multi-step complex assays that could be used really nicely with the machine automatically and allows you to collect a lot of data and again, to do what we like to call multifunctional profiling of your samples. So to conclude my short introduction of the technology, I would like to share with you this really nice lung organoid image that we obtained by staining, again, using staining protocol with our Puma system and confocal imaging. And I also would like to mention here that very soon we will release our new technical note on how to do assays with Matrigel with our Flowchip. And also in July, we will do another webinar on the same topic, going into details and aspects of using Matrigel. So please keep an eye on our announcement and register. And here I also share with you my contact information. So if you have any questions or would like to discuss your particular assays and how we can use Puma system for that, please reach out to me at any time, and I would be happy to answer your questions.
Thanks, Katya. So one of the questions from Sabrina is, do the organoids have to be preformed prior to plating, or can single cells be plated to allow aggregates to be formed?
So at this point, we use organoids or any other models that are pre-cultured and developed somewhere else, and we transfer them inside the Flowchip. We do some work on, assessing the possibility of creating them inside with cell lines, but this is ongoing work. So as soon as we have new data, we'll share it with you. But as of today, we transfer structures from outside.
Another question from Rida is, considering the size of the chamber, what is the maximum number of spheroids that can be loaded?
I would say I would comfortably use dimensions between, like I said, 100 microns and maybe 900 microns or 1 millimeter. It will pretty much occupy the full chamber, but to play it safe, I would say about 1 millimeter or so.
And I think you already addressed this question, but are you able to accommodate Matrigel grown organoids?
Yes, you can use Matrigel. We've tested, and it is compatible with between 20 to 30% of Matrigel, so that we don't block and maintain the proper function of microfluidic channels. And like I said, please keep an eye. We will be releasing some technical documentation of that soon.
Is it possible to examine the organoids under the microscope during the process?
So for now, we don't have microscope inside the machine. It only does automatic transfers and incubations, and it was done purposely because there are different systems that people use. So what you can do, if you need to examine during the protocol, you can stop your protocol, take the plate out, and then kind of resume it. Again, there are technical nuances that we can discuss offline on how you can do it, but that's the capability we have.
Awesome. Thank you so much, Katya, for the technical introduction. I will go on to Margarite and to introduce our next speaker, Margarite Matossian. She's a physician scientist who recently completed her graduate and medical training at Tulane University Medical School in New Orleans. She will be soon starting a position as a resident physician in internal medicine at the University of Chicago. Her doctorate work was under the direction of Dr. Matthew Burrow and Dr. Bridget Collins at Tulane, in close collaboration with multiple groups. She is a board member of Santé Total, a nonprofit medical organization that provides healthcare to rural Haiti. She's passionate about engaging with local breast cancer community and support organizations, and she has published several papers already in this field. And without further ado, I give to you Margarite Matossian.
As mentioned, my name is Margarite Matossian. I'm sad to be leaving Tulane, but today's webinar will be talking about work that I performed under a co-directed lab there, in which we use patient-derived organoids as translational research models to really interrogate therapeutic response and drug resistance. Of course, we always want to acknowledge those who have helped us get to where we are, both in establishing the patient-derived models that we'll be discussing in our group and in our biospecimen processing core, as well as those who have helped us to realize the applications of organoids and what we can do with these really impressive models at protein fluidics and molecular devices. Furthermore, Krewe de Pink is a local breast cancer organization who have directly contributed funds to establishing our PDX models and really helping our lab. So thank you to them, and always thank you to the patients who donated the tissue specimens. There are three major applications of organoids and spheroids that our group focuses on. We evaluate drug response using cell-derived and PDX-based models. we generate spheroids from overexpression and knockdown or knockout genes in established cell lines. And we can visualize how genetic manipulation affects spheroid growth and development, as well as other biologic endpoints. And we evaluate changes in cell culture conditions on the spheroids themselves, or the organoids. For the purpose of this talk, so I don't go on for 5 hours, I'll be discussing how we evaluate drug response using these models. Now, our group uses this highly translatable tool of patient-derived xenograft models, which has transformed the field of breast cancer research and discovery of drugs. So our workflow is as follows. We work closely with surrounding hospitals in the greater New Orleans area. We acquire patient tissue after consenting, of course. The tumor tissue we acquire, as well as adjacent adipose tissue or other biospecimens if it fits our needs at the time for experimentation. The tissues are processed through a biospecimen core facility at the Louisiana Cancer Research Center. And then we use these models as we cryopreserve for future use. We can propagate these models in mice for more immediate use. And then we can characterize these tumors using genomic sequencing or on a genomic level, as well as histologically and looking at other features. And then we can generate cell culture systems of 2D or 3D in order to further interrogate these models in our work. Our nomenclature is as follows, as you'll see throughout this presentation. We use TU-BCX and then the specific PDX model name. And then following that, you'll see T1 or T0, and that's demonstrative of the serial transplantation in a murine model, with T0 being the original tumor, and T1 being exposed to a mouse tissue one time after one passage, etc. Our group, as a breast cancer group, we specifically focus on triple-negative breast cancer, or TNBC. And what this is, it's a subtype of breast cancer that's actually an umbrella term. It's all the breast cancers that aren't encompassed by, that lacks expression of hormone receptors, so estrogen or progesterone receptor, and lacks HER2/neu amplification, which are commonly targeted proteins in breast cancer and have effective therapies. And as an umbrella term, you can imagine that TNBC subtypes encompass a variety of different cancers. And there have been efforts to further subclassify the subtype based on molecular features or behavior of the cancers themselves. TNBC itself is clinically aggressive with high rates of metastasis, recurrence, and drug resistance. And there's a higher incidence in younger women. And there's worse outcomes in patients with specific ethnic backgrounds, such as African ancestry and Latinx populations. There's an increased risk in obese patients, and there's a genetic predisposition for TNBC. Here in Louisiana, or should I say down there in Louisiana, we had high rates of mortality and incidence of TNBC, which are due to some of these contributing features. So we had a unique population that we really wanted to represent in our research to improve visibility in our breast cancer studies. Now, as we can evaluate drug response in a variety of models that are derived from these patient tissues. And I just wanted to mention here the translational hierarchy of these types of models that we can use, with the most translational being when you treat a patient themselves or the tumor within the patient in a clinical trial. That's the most translational you could get. The step before that is implanting an intact tumor, so taking a piece of a tumor, implanting it in a mouse, and then treating the mouse model or another animal model. We've historically used immunocompromised mice models, but now there's been a shift to use humanized mouse models to maintain an intact immune system. You can also treat the intact tumor in a dish as an explant. The step below that would be to maintaining the microanatomy and the cancer composition of the tumor itself as patient organoids. So you're stripping away the matrix or the structure of the tumor, or the architecture of the tumor. And then the least translational step, but also a very important area to study, are the cancer cell population. So generating a primary cell line from these tumors and then either looking at the adherent cell population or testing drug effects on spheroids with a 3D population. And I will say in our early drug treatment experiments, as we grew comfortable using these 3D model systems, we observed differences in drug response when we compared 2D and 3D systems. So this example is a mixed cell line that was derived from a PDX model 2K1. And these are a panel of targeted and more systemic cytotoxic chemotherapies. And what we observed was in some subsets, there was an obvious distinction in drug response when we treated adherent cells versus when we treated spheroids. This is a live dead viability stain with calcium AM to highlight the live cells and Pythium-3 to highlight the dead cells. And all the cells were treated at the same dose. So an interesting observation. This same pattern we observed on a genomic level as well. When we were examining PCR biomarkers with our specific drugs that we were interested in, there were obvious distinctions in drug response when we treated the adherent cells and the spheroids. This led us to draw some interesting conclusions that more translational models are needed in the lab setting to really draw accurate conclusions about drug response. And the lack of use and the lack of the number of translational models used may actually contribute to why only 3% of oncology drugs make it to clinical trials. This also showed us that drug response may differ when we're considering treating an intact tumor, or at least the microanatomy of that intact tumor, versus when we're just treating the cancer cells grown on plastic or in a dish. And then the presence of heterogeneity within the cell composition itself within a tumor may contribute to this variety in observed drug responses amongst various experiments. Using these lessons that we learned from our preliminary drug experiments, we then started to delve more into the use of organoids and applications of organoids in our research work. Many of our methods in experimental design were adapted from Alana Whelm's group at University of Utah. And she has this great schematic that shows how organoids can be used in functional precision medicine as testing single compounds or drug combinations or various drug doses. There's a high fidelity of the organoids and the PDX models to the patient tumor, where the RNA, the genomic composition of the organoids are reflective of the patient's tumor. And this helps to streamline drug response validation. And our processing workflow, a very simplistic overview, is we would digest the tumor pieces in a mixture that contained collagenase or something else to break up the extracellular matrix. Through a series of centrifugation and filtration steps, we would propagate the remaining cells in a 3D culture media in the presence or in the absence of Matrigel and generate these organoids. Or we would establish a single cell population or a primary cell line and then generate spheroids from those primary cells grown in 3D culture media. So how do organoids address this molecular heterogeneity? We understand that a tumor is not just comprised of a sheet of the same cells. So a tumor actually has phenotypically different cellular populations. And when we take a biopsy, we may only be studying one of those populations. So we can effectively find a therapy that treats this population, but we're not examining all the subclones within that tumor that may have different responses to that chemotherapy. So it's really important to remember that this heterogeneity concept and how this can contribute to drug resistance and effective drug therapies. I think I already mentioned a couple of these findings about how organoids can improve drug discovery experiments, but they're also cost effective. They require a smaller amount of tissue, and we're able to study drug effects on various components of the tumor, not just the cancer cells, but other stromal factors that contribute to tumor development. And furthermore, studying tumoroids or spheroids made from primary cells, these cells are less susceptible to the acquired mutations that established cell lines have after being plated on plastic for many years. These are images of our early organoid models. This is an unfiltered image, but one of our earliest organoid models. You can tell how excited we were to see organoids budding from our cell culture. And then when we performed a stain to examine cancer stem-like cells with a CD44 high, CD24 low population with CD44 in red, 24 in green, we really were able to visualize these organoids budding from tumor remnants. Our earliest applications of these organoid or spheroid models were to examine how drugs affected the CD44/24 cancer stem cell-like population. And then we also examined morphometric parameters of the spheres of organoids themselves, and we were able to visualize how drugs affected sphere formation and development. For the remainder of this talk, I'm going to be mentioning heavily a PDX model that we often use in our group. So here I wanted to introduce you to this model. This is TU-BCX4IC, a PDX model established from a 57-year-old white woman, stage 4B, at the time of mastectomy, high grade, a very rare histologic subtype, metaplastic, of a notoriously rapidly growing tumor. The size of the tumor at mastectomy was very large, 13 centimeters, with lymph node involvement, but no distant metastasis, probably due to the rate of growth of this tumor. And furthermore, this patient was resistant to 3 cycles of a commonly used breast cancer chemotherapy regimen, ACT, so adriamycin, cyclophosphamide, followed by a taxane. And what we observed when we took this tumor, a small piece of this tumor, and we implanted it in a mouse, was historically our average rate of tumor take. So from the time when you implant a tumor in a mouse to when you first see it start to grow, was about 150 to 200 days for some of these models. But 4IC grew within 7 to 10 days and hit maximum growth within 2 weeks. So a very rapidly growing tumor, even in the absence of Natrigel, which is often used when you're implanting PDX models to propagate their growth. And then there were presence of spontaneous metastasis throughout all serial transplantations of this tumor. So a very aggressive model. Then what we did was we were curious to see, since the patient was resistant to 3 cycles of chemotherapy, could we generate a primary cell line, and were these primary cells also resistant? Did they also exhibit a drug-resistant phenotype? For this, we generated our primary cells, and we used the NCI Oncology drug set. The panel, at the time, it was over 106 NCI-approved oncology drugs, not just for breast cancer, but for all liquid and solid tumor types. And I think now it's up to 150. And we treated our primary cells with this drug panel. And we compared our findings to an established TNBC cell line 231. And what we observed was a highly resistant phenotype in our 4IC cells compared to the 231 cells. So a very resistant cell line as well. We wanted to test some targeted inhibitors that we often used in our group, or that we were interested in at the time. And we wanted to test if these targeted inhibitors could affect even this drug-resistant PDX model. So we generated organoids from these tumor pieces. So these are passage 4 organoids, and we use a live dead stain, the calcium amethitium type 3, and we treated the organoids at a dose that we had found to be relevant in our 2D studies. And we were able to observe cytotoxicity in our organoid studies. So some of our earliest studies with these organoids were a proof of concept that we could perform these experiments. Along the way, though, we did identify some technical challenges. It's always important to evaluate how we can improve. So we did want to make sure that we had appropriate imaging techniques to visualize these really complex 3D structures. More specifically, we wanted to make sure we could evenly visualize the outer cells compared to the cells within the organoid itself, or the spheroid. And we had some difficulty in scaling up due to limited tissue availability. So we're working with a sliver of tumor or some small amounts of organoids, and it was difficult to propagate them for more further and extensive experiments. And then these longer-term organoid or spheroid experiments, sometimes going on for more than 2 or 3 weeks, were susceptible to contamination. And there were some challenges in not disrupting the really fragile organoid structures when we were changing out the media for drug treatments in various conditions. Not all organoids form a nice, tight 3D structure, and we didn't know if our cytotoxicity effects were due to traumatic disruption of the organoid structures when we were changing out the media or the drug itself. So then the Protein Fluidics came, we applied for an award with them. They introduced us to this system that allowed us to realize novel applications of our organoid studies. It was surprisingly easy to use, which I often told Evan, but it's very true. The system required only small volumes of tissues and reagents to help us combat our concerns with tissue availability. We were able to image these studies in well without having to transfer the samples to our imaging system. And then it minimized disruption to the organoids or spheroids using those microfluidics chambers. And this is all under sterile conditions. This PUMA system fits in your incubator, so it's really easy for us to use. And there were diverse applications with various biologic endpoints. So we were very happy to collaborate with Protein Fluidics. And furthermore, I just want to hammer home that it is incredibly simple to use. I'm not great with technology, so for me to say this, it must be true. But very simplistic overview, we just had to load your reagents and organoids. For our drug studies, we loaded the stain and the drug, and then an organoid. You can have multiple organoids in a well if you were interested in not having biologic triplicates, but we could have up to 3 organoids comfortably in this well. We would transfer the intact organoid. These are not generated from single cells plated in the PUMA system. All we did was we add this Flowchip and the chambers to the PUMA system. There were protocols that were already made by Protein Fluidics, and we could custom make them depending on what types of experiments we were interested in running. Really just hit play, walk away, go enjoy some dinner, come back the next day or whatever the end time point was, and image or analyze. And these images were performed with molecular devices in California, and really get these beautiful in-depth pictures of organoids. So this transformed how we looked at testing drug efficacy with our spheroids or their organoids. This is one of the earlier images we obtained with the PUMA system and Protein Fluidics. This is that live dead viability stain with some targeted inhibitors. So really just showing that you can see on a single cell level how drugs affect organoids, or cells that comprise an organoid. We used this technology. At the time, we were finishing a paper on how to characterize that aggressive PDX model that I mentioned, 4IC. And what we had done was we had taken the primary cells that we had generated, we treated the adherent cells with paclitaxel since the patient's tumor was resistant to a taxane therapy, and we hit with physiologic doses of paclitaxel. We did find that there was a very minimal response of the adherent cells. And we wanted to really translate these 2D findings into the patient. There was a barrier or a gap there. And Protein Fluidics was able to help, and the PUMA system was able to help us really translate these findings better. We performed the exact same live dead stain, but using plated spheroids. And we were able to observe that at much higher doses than what's physiologically relevant, that the 4IC cells were still resistant, or now the spheres. So further demonstrating that our PDX model had a highly drug-resistant phenotype. And molecular devices using analyses masks helped us to quantify these observations and really find an inhibitory concentration value. These are videos. I'm not sure if they'll play, but we'll try anyway. But they're able to capture the depth of these organoids, and you can really see changes to drugs, changes from drugs on a cellular level within the tumor and in the outer cells of the tumor. So really amazing images. We were also interested in other biologic endpoints other than viability. So this was a test experiment to see if we could stain with a cell adhesion molecule that we're highly interested in, ecadherin, and that cancer stem cell marker, CD44. So this is with paclitaxel and the 4IC spheroids, and we obtained these beautiful images. You can see a high concentration or a high presence or expression of ecadherin, and how the paclitaxel treatment affected biomarkers expression as well. Importantly, we were able to capture these observations on a large panel of drug doses, so we could really visualize and quantify these expression changes using very minute changes in drugs. So you could capture a very good dose-dependent response. And one thing I also wanted to mention that was very important for us is I mentioned the fragility of these organoids that we had encountered. A group at MD Anderson actually developed this technology of the nano shuttle. And what it does, you can coat cells with these iron nanoparticles and use a magnet to help maintain the structural integrity of very delicate spheroids or organoids. This is an out of focus image, but an in focus on the nanoparticles. You can see that they help hold the organoid together. And the Puma system has this technology where it's built into the staging system. So it helps pull down these organoids, and you can really use this technology for these fragile structures. So really important to us. This doesn't just help with organoids, but also with spheroids, or cells that are known to not generate structurally integrous spheres, such as MDA MB231 cells, which at baseline have low levels of cell adhesion molecules and proteins, which make it difficult for you to form spheres that actually form more aggregates. But using the nano shuttle helps keep these cells together so you can perform drug treatment experiments. Another obstacle that I want to mention that organoids are very hopeful for is drug resistance. This concept of sometimes within a tumor cell population, there are clones that actually will not be affected by any chemotherapy, as in our very aggressive model. So some of these subclonal populations, no matter how many times you treat with the chemotherapy, the cell population itself isn't really going to be affected. So this results in a highly resistant tumor population that's allowed to continue growing despite chemotherapy use. Or you can have induced chemo resistance or acquired chemo resistance, where some of those subclones within a tumor that aren't affected by chemotherapy get a partial response. And then over time, those subclones that aren't affected by the chemotherapy can grow and populate and cause tumor recurrence or cause tumor growth despite the chemotherapy regimen. So a really important concept that prevents effective therapeutic regimens. And organoids can be used to interrogate the subclonal cellular responses to these therapies. So as I mentioned before, there are a simplistic overview of drug resistance with these populations is within a tumor itself, you have some cells that will respond. You have some cells that might evade the therapy. And you have some cells that might grow despite the therapy and have metastatic capabilities and cause metastasis in the patient. And we can use our organoid system to address these concepts in the laboratory setting. So a couple experiments that we were currently working on when I left were you can take organoids derived from a PDX model or a tumor that's actually drug resistant, as the aforementioned aggressive PDX model, for example, the 4IC model. Or you can generate subclones from these drug-resistant tumors that represent the subclonal populations. If you hit the drug-resistant tumor with a targeted inhibitor, you can, and the inhibitor is effective, you can say that potentially the signaling pathway is a possible mechanism to overcome that drug-resistant phenotype. Or you can treat the various subclones and interrogate why some subclones have a more robust response to these targeted therapies in the specific signaling pathways, while there's lack of response in other subclones. So using these organoid models, you can really interrogate drug resistance mechanisms. You can do this on a larger scale using a target probe or a drug set to really interrogate what you can compare. You can use the target probe or the drug set, hit organoids with these therapies, and then compare what's a hit and what's a miss. So which therapies were effective and which therapies lacked a response. And you can identify by comparison which downstream targets or signaling pathways you want to pursue as potential mechanisms of resistance, or those that induce a lack of response. We are very excited about the PUMA system, and I know when I left the group, there were many continued experiments that we were excited about, including evaluation of secreted factors in the cell culture media, because you could harvest the supernatant from the media in the PUMA system. You can examine the presence of secreted factors or exosomes within those cell culture conditions. We've been working on trying to extract RNA or protein from organoids growing in the PUMA system by flowing in RNA lysis buffer, protein lysis buffer, and then just extracting out the resulting RNA or protein. And then, as mentioned by Katya, currently we're working on how to use Matrigel in these PUMA systems. This is incredibly important for us because some organoids really require the Matrigel to grow. So at this point, I'll stop sharing and see if anyone has any questions. But thank you for your time.
Thank you so much, Margarite and Katya. So I have a bunch of questions for both of you. So let's start with Margarite. So do you quantify CD44 intensity as a whole or average to remaining volume?
We can quantify, so to be very frank, molecular devices perform the quantification of CD44. So I think that question we would have to specify with them, but based on our understanding of the quantification, it was as a whole. So, but that's a question that I can get back to you, but it's more specific for molecular devices.
Okay. So Katya, one question for you from Nina is, are the plates compatible with confocal imaging?
Yes, the frame and these flow chips that are sitting inside the frame, the dimensions of the whole system resembles, like I said, a standard 384-well plate. So it is compatible with just regular manual microscopes, or if you would like to use in high content automatic imager scanners, you can do so as well. Yes.
Okay. One technical question for Katya is, how do you transfer the spheroid organoids to PUMA system, considering that the fact that the media volume is so small?
Yeah, it's probably one of the biggest challenges because it's a really delicate procedure. So we have developed guidelines. It's really hard for me to tell you, in very short time how exactly we use it. But we have a detailed guidelines outlining the procedure, how we do it. But briefly, yes, we do it in a very small volume. So we really depends on culture system that you use before for your organoids or spheroids. We transfer them in about really between, I would say, 2 and 5 microliters to load them into the chip. So it is very low volume.
Okay. So another question for you, Margarite, is, so now what do you think is the next step, like from when you are collecting the supernatants and samples and the continued work with the TNBC project? What would be the next steps?
Oh, there's so many. It's hard to limit yourself to one or two, but there's been a new wave of interest in drug resistance. So looking at subclonal populations is definitely of interest to us. I know some members in our lab are looking how if you manipulate cell culture conditions by using, various treatment strategies in the cell culture media itself, how does that affect sphere formation, organoid, various endpoints you can look at. We can examine other biomarkers I didn't mention in this presentation, but obviously there's many other biologic endpoints that you could be interested in. You can look at mesenchymal or more epithelial populations of cells and how those are affected by various drug treatments. I don't know when to stop. The metabolomics of cell populations. Yeah. You can look at cytokine profiling, how drugs affect those. So there's endless possibilities, but I do want to emphasize that the PUMA system really helps to streamline those processes and those experimental designs. It makes it less complicated.
Okay. So one of the questions from Andy is, there's a claim that RNA can be harvested from organoids in here. Is that feasible when each chamber only fits one organoid?
Yes, that's something we're working on, right? So we're working on optimizing the threshold, the level of threshold of RNA that we can obtain from these organoids. These are experiments we haven't dove into yet, but we were considering starting in collaboration with Protein Fluidics.
So another question.
May I add just one
please.
thing to the, yeah. So although today we shared with you data in showing really that we can do one organoid and get data from one, there's really different options. So if you would like to put few of them, again, depends on the size, you can do so as well. It all depends on the technique and lab practices that you use in your lab. But if there's a requirement to get more of them, to deal with thresholds, that's a possibility too.
Okay. So another question was, since the volume is so low, have you been looking at, say, exosomes in the supernatant? Is that something that you would look at?
Again, a proposed experiment, but one that we have not optimized yet. But
okay.
Hypothetically, you could, depending on the level of threshold of your experiment choice, right?
Yeah.
So, but something we'd be interested in.
Yeah, and Andy, I do want to mention, similar to what Margarite mentioned with regards to the RNA, we have done experiments where we've shown that you can do lysis within the chamber. So at the end of the experiment, you can pull the Flowchip holder out, transfer in, say, a lysis solution. It could be lysis for metabolomics or for transcriptomics or genomics. And you just have, the system would do the automated transfer, and you could lyse and then take out that for the next, and take it out, the supernatant, the lysate rather, for the next downstream process. So I'm just waiting to see if there are any more questions. Please feel free to send them into the chat. And if there are no more questions, we will go on and say goodbye and thank you. But we will stay on and answer your questions by chat. But we can go ahead and end the session if there are no more questions. Thank you so much, Margarite, for your time today, telling us about your amazing research that you have done so far with triple-negative breast cancer. And thank you, Katya, for the applications and for telling us about the Tumor System workflow. I appreciate all the attendees for today, and thank you for your time.
Thank you.
All right, goodbye.